Hello everyone, welcome back to the Center of Excellence CET Crash Course. In this session, we'll move on to the chapter Molecular Basis of Inheritance. So we'll start with the first question without wasting time. So the first question is, the results of the following experiment reaction carried out by the Avery et al. On Streptococcus pneumoniae has provided conclusively that DNA is the genetic material. So they were telling that which is the experiment done by the Avery et al. Avery et al. That is Oswald Avery, Maslin McCarthy, and Colin McLeod. So these three scientists have continued the experiment of Griffith's transforming principle. Griffith has done an experiment on Streptococcus pneumoniae by using the two different strains, that is R strain and S strain. What he's seen is the conversion of the R strain, that is non-virulent strain, into S strain. So according to the Griffith's transforming principle, non-virulent R strain converted into virulent S strain when it was mixed with heat killed S strain. So the same, but he could not give what is the theory behind it. So the Avery and Avery and the other, they have worked on the same experiment by using some of the digestive enzymes. So Avery et al. They have used the digestive enzymes. And by using the digestive enzyme, they have digested the varied macromolecule, mainly protein, then lipid, then the carbohydrate, then RNA and DNA. So they have used different digestive enzymes. For example, for the protein is digested by enzyme proteases. Lipid was digested by lipases, carbohydrases, then RNases, okay, 1 minute, again got stuck over here, and RNases and also DNases. So they have used different digestive enzymes. So when they have used all these digestive enzymes, okay, whether it is protein, lipid, carbohydrate, or RNA and DNases, they have seen some of the variations between these enzymatic activities. Okay, so RNases and DNases. So when they have used all these enzymes, that is from protein up to DNA, there will be the transformation of non-virulent strain into virulent strain. So when they have used DNases, there will be no transformation. They have not seen any transformation they have used when they have used DNA. This is why it digested complete DNA material. Okay, so it digested the DNA material. As a result of that, when they have used S strain and the R strain, so they have used live R strain, and mainly they have used the S strain macronucleus, macronutrient or the macromolecules, and these macromolecules are mixed up with the live R strain. So that time what happened, the conversion was taken place. When they have not seen the conversion, that is mainly when they have used DNA. So the answer is live strain R strain plus DNA from S strain plus DNases indicated that there will be no transformation in the absence of the DNA, because when they have used all the other things, DNA is present. When they use proteases, protein is removed, remaining macronutrients are present or macromolecules were there. So when they have taken the lipases, lipid is digested where the other macromolecules were present. Okay, so when they have removed DNA, so DNA is digested out and remaining macromolecules were there, and they are not affected. Okay, they have affected towards the transformation process. Okay, so there will be no transformation. So the answer is live R strain bacteria. Let's move on to the next question. Which of the following events would occur in lack of around E. coli when the growth medium takes place in a higher concentration of lactose molecules? So as we know that if you consider lac operant model, so they will be having different um, we have I gene, we have P gene, we have operator gene, we have Z, Y, A gene, right? So these are the structural genes. When I gene produces a repressor protein, and this repressor protein readily goes and binds to operator gene. Remember that repressor protein goes and binds to operator gene, and now this repressor protein inactivated. So the protein, so if it is goes and binds here, so there will be no transcription. So there are no transcription when they bind, because the polymerase enzymes cannot move due to the blockage of their way, right? So when the repressor protein binds to lactose molecule, means when in presence of lactose molecule the transcription process initiated, or there will be the transcription was took place. Transcription is seen. Okay, so that is only when the repressor inducer complex, the protein binds to lactose molecule. So what do you mean by this? This is forming repressor inducer complex. It is formed repressor inducer complex. Okay, so now the answer what is given is the structural gene fails to produce polycystonic mRNA. See, what do you mean by that? There is a lactose molecule is present in the concentration is more, means it containing the media where we have a lactose molecule. So when there is a lactose molecule, structural gene fails to polycystonic mRNA. When there is a lactose molecule, polycystonic mRNA, the transcription takes place, right? So mRNA is produced. So this statement what is given is wrong. So the repressor protein binds to RNA polymerase and prevents translation. Repressor protein attaches to the promote sequence and depresses of the operator. So all these three statements what we have, that takes place in the absence of lactose molecules, in the absence of the lactose molecule, not in the presence of the lactose molecule. So now in presence of the lactose molecules, what happens, there is a formation of repressor inducer complex. Inducer molecule binds to repressor protein and RNA polymerase binds to promoter sequences. So this is the correct answer. Let's move on to the next question. So statement A, the primary transcript produced in eukaryote is translated without undergoing any modification or processing. Statement B says the hnRNA in humans has exons and introns. Okay, so as we know that when transcription got over, after transcription, so after transcription, if we consider an mRNA, so this mRNA usually contain or the eukaryotic protein or eukaryotic genes are called as split genes. Why they are called as split genes? Because they contain exons and introns, alternative exons and introns. Right? So they are called the split genes. So the hnRNA. So this is what you call is your hnRNA, that is heterogeneous nuclear RNA, heteronuclear RNA. Okay. So now hnRNA in human has exons and introns. The statement B is correct. So now statement A says the primary transcript produced in the eukaryotes is translated without undergoing any modification or processing. So we cannot do such kind of things, because if we go without any modifications, we have non-cystonic part of the DNA. So this is the coding part, this is non-coding part. So we require all coding part together. So after transcription, hrna will go to the post-transcription process. Post-transcriptional process, and in the post-transcriptional process it undergoes splicing, that is removal of introns and joining of exons. Then we have capping, then at the end we have your tail. Right, so we have capping and tailing. So these are the three processes. So here they are going to cut the cut the introns, and exons are joined. Where in case of capping, guanosine 3-methyl guanosine, okay, trimethyl guanosines are phosphate, methyl guanosine triphosphate are added to 5th prime carbon. So where polyadenylate residues are added to 3' carbon. After all these processes, the mRNA that is produced, 5' carbon to the 3' carbon, so this is produced, is called messenger RNA. So this is produced after post-transcriptional process. So this is what is given, the statement, that is A, transcripts produced in the eukaryote is translated without undergoing any modification. This is wrong. So statement B is correct and statement A is wrong. So the answer is C. Let's move to the next question. Some of the steps of DNA fingerprinting are given below. To identify the correct sequences from the option given. So, electrophoresis of DNA fragments, hybridization of the DNA probe, digestion of the DNA by RNAs, restriction endonucleases, autoradiography, blotting of any fragments into nitrocellulose membrane. So, what's the first thing? First, we are going to, suppose if we have a DNA and we are going to cut the DNA into small fragments, so by using your restriction endonucleases. So this is the first thing, we are going to use the restriction endonucleases. After restriction endonucleases, we will move on to the electrophoration. After electroporation, we are going to copy those DNA. So in case of electroporation, what happens? Movement of DNA with respect to size. Okay, so from larger to smaller. So we'll be having a small at the lowest part. So after this one, what we do? We are copying this DNA to nitrocellulose media. Paper, nitrocellulose sheet or membrane. Okay, so this is what we call as a southern blotting. We have western blotting, southern blotting, northern blotting, all those things. Okay, so now this is the next one. After this, we are going to hybridize it with the DNA probes to understand whether the DNA what we are looking into having the same or different. Okay, so then hybridization. So this is first one. After this one, we are moving into the electrophoresis. Then electrophoresis, we go on to this one. Then hybridization. At the end, we are going to visualize it under UV radiation or X-ray diffraction. Okay, so that is this one. So starting with a C, then we'll be having A, then we have E, then B and D. So the answer is A. Let's move on to the next question. If E. coli is allowed to grow for 40 minutes in a medium containing N15, then the number of N14 and 14 containing DNA would be. So they are telling that they were growing that the DNA for 40 minutes in N15 media. So it means we have a DNA here. So now this DNA what we have is N14. So I'm going to put it into N15 media. So when I put them into N15 media, semi-conservative replication occurs, and after the semi-conservative replication, the new DNA what is formed is 15. This is N14 and this is N14. Right? So we'll be having the hybrid DNA at the first. So we'll be having the hybrid DNA at the beginning. So this is the first generations, if I consider. So again, I'm going to keep it for the second. Let's go mention about 40 minutes. 40 minutes is 20 minutes. If we consider each generation, they have mentioned about E. coli bacteria. E. coli bacteria will multiply every 20 minutes. So after two generations, the first generation, this is the second generation. So now this generation, if I consider. So I know that this is 15 and this is 14. So the new stands what is producing, both of them are 15. So here, so this is a hybrid and this is N15 stand. So but we want N14 stands. So there are no N14 stands are produced in when you're putting that into N15 media for the second generation. So we cannot see either we will be having the N15 and 15 strands, or we'll be having hybrid strands. Okay, so the answer is 0. Let's move on to the next question. The polymerization of the DNA nucleotide during the synthesis of the lagging strands occurs in, if you consider the synthesis of the DNA. So this is the DNA strand. So now this is the 5th prime carbon, this is the 3rd prime carbon. So this is the 3rd prime carbon and this would be the 5th prime carbon. So now this is what called is replication fork. So this is a replication fork. So towards the replication fork, that is from the 5th prime carbon to the 3rd prime carbon again. So this strand is called continuous strand. where away from the replication fork, so from the 5th prime carbon to the 3rd prime carbon, this is not actually continuous, this is lagging strand or discontinuous strand. Okay, so this is what is called Okazaki fragments or your lagging strand. So remember that whichever it is, whether it is RNA translation or it is DNA replications, whichever you take, always the strands, the new strand always formed from 5th prime carbon to 3rd prime carbon. It is not vice versa. It is not from 3rd prime carbon to 5th prime carbon. It is always to 5th prime carbon to the 3rd prime carbons. Okay. So the answer is 5th prime carbon to 3rd prime carbon. The anticodon found on tRNA for tryptophan amino acid is, you know what you mean by anticodon? If I consider your tRNA. So if you consider this tRNA, so in the tRNA, this portion what you called as your anticodon arm. Okay, so the portion what we can see over here is anticodon arm, and now this anticodon arm with respect to your mRNA, what you have in case of mRNA. So if it is amino acids, what we have over here, so the amino acid what is present over here is tryptophan. Then codon for the tryptophan is UGG. So you can make out tryptophan is the only tryptophan is the only amino acid which are having only one codon. Okay. So if I consider UGG, then obviously the anticodon arm consists of, okay, the anticodon arm consists of the opposite to the one what we have. You will be having A, then we have your C, then we have C. So the answer is ACC. Okay, so the ACC in case of tRNA, ACC is the answer for the tRNA, okay, of the anticodon in case of your tryptophan. ACC is the tRNA space. Okay, so let's move on to the next question. In case of which of the following amino acid is coded by the single codon? I have already explained to you about that, which is the codon contains your single codons, as we have seen in case of in this particular codon table. Okay, can make out, so phenylalanine contain 2 codons, so leucine contain 2 codons, so maximum codons are also present here. So where the tryptophan is the only amino acid contain only 1 codons. So we have a stop codon, and these stop codons are responsible to stop the translation process. So the answer is tryptophan. Let's move on to the next question. In prokaryote, the transcription of DNA is initiated with the help of, if you consider the transcription, so this is a transcription unit. So usually the transcription unit contain the initiator part of the DNA or initiator, okay, initiator gene. So this is what we have is a structural gene, and we have terminator part. Okay, terminator sequences, initiator sequences, terminator sequences, and will be having structural gene. Okay, so now, so they have asked, in prokaryotes the transcription of the DNA is initiated with the help of. So usually the transcription initiated when the uh, RNA polymerase attaches to your promoter gene. Okay, initiator code. I mean, not initiator codon. So it is to the initiator sequences. That is mainly for the promoter gene. So now this promoter gene where it is initiated. So the sigma factor will attaches and initiates the transcription of so this should be 3. So initiate the transcription of HNRNA. Okay, so from 5th prime carbon towards your 3rd prime carbon. So this will act as template strand. When it reaches to the terminator sequences, rho factor will come and attaches to the RNA polymerase. As a result, it just detaches and mRNA or the hnRNA will released into for the post-transcriptional processes. So the answer is sigma factor. Let's move on to the next question. According to the Human Genome Project, the total number of genes in the human genome is estimated as 30,000. The number of genes present on Y chromosomes are. So the least number of genes are present on the Y chromosome. So the lowest number of genes are present, that is 231. So where the chromosome number 1 contain the highest 2968 genes present in chromosome number 1. Okay, so there are about 802 gene found on chromosome X. Remember that. Okay, so the answer is 231 genes that are present in Y chromosomes. Let's move on to the next question. In crime investigation, the investigating officer collects the different biological samples from the crime spot for the DNA fingerprinting analysis. Which of the following sample is not helpful in the analysis? So when they have visited the crime scene and they are collecting the evidences from this crime scene to go for the forensic analysis, okay, they found they have got some skin cells, they have got some erythrocytes, they have got some semen sample, they have got some hair follicles. So if we consider, we can use all these cells except erythrocyte, we can use all these cells for your genetic engineering. Why? Because these cells have nucleus. So when they have a nucleus, they will be having the DNA, and we can use that DNA for your genetic fingerprinting. Okay, genetic engineering. So that is mainly I will speak in terms of the genetic fingerprinting. Okay, so but I cannot use the erythrocyte. Why cannot use erythrocyte? Because matured erythrocytes are matured erythrocytes. What we have, they are enucleated, so they do not have any nucleus. When there is no nucleus, there is no DNA, then we cannot use it for your genetic fingerprinting. So the answer is erythrocytes. Okay, let's move on to the next question. A mature mRNA consists of 900 bases without any stop codon in between. Calculate the number of amino acids coded by the mRNA during the translation. So we have a 900 codons, 1, 2, 3, 4, 5, 6, 7, 8, 9, so on, moving into the 900 bases. So this is a 900 bases. So now without any stop codons. So they have told without any stop codons. If I say, if I am taking all 900, so then I can take, there will be formation of 300 amino acid sequences. The sequence of the amino acid would be 300, but they have mentioned there will be no stop codon in between. So obviously we have to get 300 amino acids, but they haven't mentioned whether the stop codon is present. As it is mentioned, it is here matured mRNA. So it is mentioned matured mRNA. So obviously you will be having the last three bases. What do we have? UAA, if I consider. Okay, so the last three bases. So this is the 901. So the last three bases are acting as, or UGA, okay, are acting as stop codons. Right, so if I am considering these three are the stop codons, then we cannot produce 300, then we will produce one less amino acid. So the answer is 299 amino acids are produced during the mRNA translation. Okay, which of the following nitrogen bases found only in DNA? So if I consider nitrogen base, if I consider nitrogen bases, if I consider in case of the DNA, and if I consider in case of the RNA, so I have in case of DNA, I have 89, I have guanine, cytosine, and thymine. So where in case of RNA, I have 89, I have guanine, I have cytosine, and I have uracil. We do not have thymine in case of RNA. So all the other three are found in case of DNA also. So the thymine is the only one which is found in case of DNA, where in case of the uracil is the only one which is found in case of RNA. So we do not have uracil in case of the DNA, and we do not have thymine in case of RNA. So found only in DNA, that is the nitrogen base found only in DNA is thymine. Okay, so let's move on to the next question. We have an choose the four. VNTR. So we have to match the following here. So we have given a set of different characteristics here. VNTR, we are variable number tandem repeat. Okay, so where it is usually used in case of DNA fingerprinting, right? So variable number tandem repeat is used in case of DNA fingerprinting. Introns and exons are removed using the splicing, and dystrophin is the largest gene, where you will be having around 1.5 lakh base pairs. Okay, so the largest gene that contain the base pairs, so the P, and satellite DNA is a bulk DNA. Okay, so bulk DNA, satellite DNA, because mini satellites or microsatellite are the DNAs which are present in our DNA, repeatedly present in our DNA sequences. A bulk part of the DNA is made up of the satellite DNA, which are of the repeated sequences. So the answer is QSPD, QSP, and PR. Okay, sorry, no, there will be no D here. So obviously I'm getting confused over here. Okay, fine, QSPR. So the answer is yay. Fine, let's move on to the next question. Okay, in a 3.2 kilo base long piece of a DNA, 820 bases are found. So what would be the number of cytosine bases? So they are mentioned, the given is, we have a DNA sequence of 3.2 kilo base pairs. So as I know that 1000 base pairs is equal to 1 kilo base pairs. So now if I consider 3.2 kilo base pairs, means if I convert this one into base pairs, that is around 3200 base pairs are present. Of these 3200 base pairs, they are mentioned 820, what do we have is 89. Okay, if we consider we have 820, 89, if you consider 820, 89, then definitely 89 bonds to thiamine with the 2 hydrogen bonds. So obviously we have 820 thiamines. So total, if I consider the sum of this 89 and thiamine, if I consider, that's about A plus T. If I consider, that's about your 1640 base pairs. Okay, so now 1640 base pairs of 3200 base pairs are 89 and thiamine. So remaining what we have, that is your cytosine and guanine. So the remaining is cytosine and guanine, that is around 1560 base pairs. So now we know that is C plus G is equal to 1560 base pairs. So obviously this will be equal. So C is equal to G, as we know that C is equal to G. So if C is equal to G, then obviously we can divide this 1560 by 2. We will be getting 720 base pairs per guanine or cytosine. So it is not 720 base pairs, so it is 1 by, so 780 base pairs. Okay, so we'll be having 780 base pairs, which will be the answer. Okay, the answer is 780 base pairs. Let's move on to the next question. In 125 amino acid sequence, if the codon for 25th amino acid is mutated to UAA, so UAA, as we know that it is stop codon. So if 25th amino acid codon is mutated to the UAA, that is stop codon, obviously 25th amino acid is not formed, so the amino acid chain is 24 amino acids. So the polypeptide chain of 24 amino acid is present. Okay, let's move on to the next question. The codon UUU and UUC codes for phenylalanine only. This feature of the genetic code is called. So usually the amino acid, single amino acid may have different codons. For example, if I say phenylalanine contain UUU and UUC. Leucine contain UUA and UUG. If I consider proline, we have CCU, CCC, CCA, CCG. So one particular amino acid consists 4 codons, right? So this indicates, or this is what we call as degeneracy of codons. It means one amino acid may contain or may have many codon, but one codon codes for only one amino acid. So this is called as degeneracy or degenerate nature of the codon. Okay, the answer is degenerate of the codon. So where this is unambiguous, this is what is called unambiguous, where one amino acid, so one codon codes only for proline. So this codes for only for proline, CCA only for proline. So it will not codes for leucine. There is not coding for the leucine. This CCA codes only for proline. Okay, AAA codes only for lysine. So this is what is called unambiguous or non-ambiguous nature of codon. Okay, let's move on to the next question. E. coli bacteria grew in 15 NH4Cl medium for several generations are allowed to grow in NH4Cl of containing the normal nitrogen. So after 2 generations, the bacteria are isolated from the bacteria and DNA bacteria centrifuge to cesium chloride. The results of density gradient DNA is, So as we already discussed in the last previous question, so they have taken the DNA and kept it in the NH4 media. So the NH4Cl, this is the growth media for this generation. So this is the first generation, the second generation. So the first 20 minutes will be having only hybrid DNA. So only hybrid DNAs are produced, where in case of second generations we have light DNA and also hybrid DNA are produced. So there are no heavy DNAs are produced. Okay, so either we'll be having light DNA or we'll be having hybrid and hybrid DNA, not all, and hybrid DNA. So both hybrid and light DNAs are produced.